Good afternoon, everyone, and welcome to this webinar. I'm very happy to share with you our view from panels to exome or even to whole genomes for sequencing hematological malignancies. And I have to disclose on the first two slides that the opinions expressed during this presentations are my own and may not represent the opinions of Illumina. Any use of Illumina's products described in this presentation may be uses that have not been cleared or approved by the FDA or any other applicable regulatory body, and Illumina is compensating me to present this webinar. And further, as you heard before in the introduction, I'm the part owner of the MLL, and I receive honoraria from companies where I give talks as well. I'm a member of some ad boards, as you see here. Now, let's jump into the scenario of hematological diagnosis. And for those of you who are working with this, that's not new, but for others, I just want to point out at the beginning of this presentation that today, the diagnostic techniques we used to diagnose leukemias and lymphomas at diagnosis or for follow-up or at relapse have been combined and are still combined by using cytomorphology, histology, cytogenetics, accompanied by FISH or by FISH alone, immunophenotyping or immune histochemistry, and a huge and increasing number of molecular assays in our laboratory and in many other laboratories in the world. Now, having said that and getting to the point of this presentation, these techniques are more and more accompanied or substituted in some cases as well by molecular-based assays, and within them, the readout is far away from the phenotypical readout of a microscopical picture from a blast of a leukemia, but we will finally are left over with a readout that only is combined from 4 letters, A, C, G, and T. And to demonstrate to you our experience over the last 12 years, the next slide showed you in a circus plot the different methods that we apply, starting with cytomorphology, cytogenetics, FISH, immunophenotyping, and molecular genetics. And what you can see here is that all these techniques are in most cases combined of a specific combination with the others. So for some diseases at diagnosis, we use all of these different approaches. For others, only 1 or 2, following the guidelines for diagnostic use in hematological diseases. But what also is very clear on this circus plot is that those cases that only will be investigated by molecular in blue have the highest number. That means that assays that are used here that only need 1 of the 5 different approaches are molecularly based more and more, and the others are normally coming in combinations with them. Now, what does this mean for the gold standard of diagnosing leukemias and lymphomas today? This is the new cover of the updated WHO classification of tumors of hematopoietic and lymphoid tissues from this week or the next week, because this book is now available or will be available very soon. And the cover again demonstrates that so far we have macro, we have micro, and we have some more techniques that refer to chromosomes or FISH or even on flow and scatter diagrams. But at least right now on this cover, for this book, that will be the gold standard in the next years, so far there is no molecular readout shown. Now this is different if you look to those 2 papers that have been published last year already, before the book is out, to help us to classify those leukemias and lymphomas state of the art. And these 2 papers published in Blood last year, the myeloid neoplasms as well as the lymphoid neoplasms use already a lot of molecular markers that are necessary to classify according to WHO rules. And these are the markers according to their number. They were mentioned in these two papers that have been used to describe the respective diseases in these two papers in combination. And the number of molecular structures, that means mutations, translocations, as well as expression, is now 78. That means if you are going to go for any kind of diagnosis following the updated and recent WHO classification systems, you have to have in-house a lot of molecular tools and assays to really be able to classify those diseases. Now, having said that, the molecular genetic approach in hematology today is depending on two different workflows. The first one, starting with the malignant cells after ficol or lysis, that is using DNA, and the other one is using RNA. So if we have the technique like NGS, we are able to link the patient and his respective DNA or RNA with a patient-specific index, a multiplex identifier, for example, to run one sample and one patient with a lot of other patients in the same experiment, in the same assay. And then the question, and this is one of the questions of this talk, panels, only genes, exomes, genomes, where do we go? Where are we standing right now, and where will be the future in the next, let's say, 5 to 10 years? Now, we are using now, and I will show you in detail quite soon, a lot of panel sequencing. But we also use targeted RNA sequencing, and all these assays are based on NGS. But this is only the experiment itself, and the software tools, and I will address this as well, are quite a very important aspect in the complete use of NGS for research as well as for routine. Because if you are going to use techniques like that with a lot of patients per run, you finally had to report. You have to send this report to a doctor. He has to understand the report. He has to explain the results to the patient. You want information on prognosis as well as, if possible, recommend the best treatment. Let's say the precision medicine as one of the most important words right now we intend to follow. Now, if you want to do all that, you need a lot of automation in the workflow. And this is what we do. Starting from the lysis and the storage, the DNA or RNA extraction and the measurement of the concentration, maybe storage again for the DNA for the next night, the library preparation as well as quality checks again. And finally, after all that, the sequencing can start. The whole process, as we set it up over the last 10 years, is linked to our intranet sampling data in our LIMS system. All samples are tracked for internal control by barcoding. This is also true for all biologists, bioinformaticians, as well as technicians, that the use of the samples at that time when they handle the sample is tracked as well. And we have a lot of automized bioinformatic tools to finally get to the integrated reporting of all the methods for the respective patient and the doctor. Now, as I said, we use panels. The panel we are now using in our daily routine is called heme panel, and we decided to include those all over 63 genes that cover a panel for myeloid as well as for lymphatic genes in one assay, in one approach. The reason to do this is that we don't want to use so many different approaches, so many different assays per day per patient. And the other reason is that many patients, especially in the reference laboratory we are running, that serves a lot of patients in Germany, they are not quite sure what the patient has. So the question may be unclear anemia. So if you just address, let's say, 3 or 4 or 5 genes from the myeloid panel, you may miss the BRAF mutation that would demonstrate to you that the unclear anemia is caused by a hairy cell leukemia. And so we decided more than a year ago to put all these genes in one approach, in one assay. And the question now is, if we sequence all of them for the respective patients where we know already it's a myeloid or a lymphatic disease, or we don't know where we go, how we go forward after the sequencing. So we have a specific matrix that is automatically preparing the results from the sequencing for the bioinformatician and especially for the molecular biologists that have to look into the respective findings. That means the genes are from top to down. On the left, there's 63 genes, and on the left quarter, these are the lymphatic diseases. On the right down, it's the myeloid diseases. And we, first of all, looked into the respective diseases and the respective genes of interest for these diseases before we look into the other part of the complete matrix. And then we follow a specific color-coded system. That means, irrespective of what was the question, what gene should be investigated, I have to admit that this will be the only thing we will finally be reimbursed for. But irrespective of that, having the patient's health and hopefully cure in mind, we report all in green automatically to the doctor. If it's mutated or if it's wild type, it doesn't matter. We report if clinically relevant and mutated, the ones in yellow, we report in orange if the only mutated genes, and we will not report if it's not relevant for the question and that was only wild type. That means that we have a completely, more or less, automated preparation of the report, and the molecular biologists and the bioinformaticians have to go to finalize it. Now, let's see for the numbers we did using different panels in the last 2 years. So before we had this heme panel in the third row, we used a myeloid panel with only 26 myeloid genes. We applied this more than 4,000 times. The second panel that only includes 3 genes for myeloproliferative neoplasms. This is JAK2, pyreticoline, and MPL. We have used now more than 12,000 times, and the heme panel more than 6,000 times over the last 2 years. Summing up, for more than 500,000 genes that were investigated by using panels and the respective workflows and NGS instruments. Now, having said that, I have to tell you that this complete workflow is organized and controlled in a pipeline that controls all the different steps, starting with the DNA isolation and quantification, having the instruments in place, then the data analysis, the quality management, amplicon check, coverage reports, sequencing performance, and then finally goes to our learning database. I will show you in a little minute better. And having this in-house, the very important thing is now, and this is really the most complex task from our perspective, using NGS today in routine as a maybe diagnostic tool or also in research, because it is difficult to accurately automate this process. So you see here data that are publicly available, and these databases are very heterogeneous and sometimes even inconsistent or even contradictory with the data that they include. Thus, there is no simple algorithm to do the functional classification of a variant or a mutation. We use a multitude of sources to make the call, carefully considering each piece of information. Most important is the lab's own database, because we saw a lot of different cases over the last years, all from the field of leukemias and lymphomas. And we also can include the patient's clinical course and the orthogonal information like cytomorphology or chromosome banding analysis or immunophenotyping, and potentially the validation of mutations where we are not quite sure what they mean by germline controls. So each variant is checked against presence in public variant databases such as Convic or ClinVar, or for somatic mutations in dbSNP for known SNPs. In addition, mutations impact prediction tools are used to calculate the severity of an impact of variants to distinguish between pathogenic and benign. Now, I want to show you one of the problems we are facing by doing so. Now, here are the top 30 genes over the last 7 years that we investigated in our hematological malignancies. Starting with TP53 or RUNX1 as a 3b1 and going to the right side to CSF3R. And blue color-coded is the number of mutations we saw in the respective gene. This is up to, let's say, 3,500 in P53. Now, this is what we did in-house here and what we patient per patient and gene by gene finally put into our report. If you then go to ClinVar, as shown on the next slide, and look for the top 30 variants that you can depict on ClinVar, gene by gene, you will be surprised that, at least from the hematological point of view, you are not seeing so many genes I showed you before, because this information is much more based on oncological diseases, cancers, solid tumors. And if you ask yourself about the overlap of our top 30 and the ClinVar top 30 genes is 0. That means we are very often have to go back to our own database to see if there's a new finding. And as databases outside hematological world show a lot of these genes that we are not normally seeing in our diseases has to be taken into account. Now, during routine workup, we classify all calls from the variant caller in a 3-tiered system we use in-house. Single nucleotide variants with a high frequency in the population are classified as benign. We use frequency data provided by Genome AD project, the successor of EXAC. Somatic variants are either known entity-specific hotspot mutations, protein truncating variants like frameshifts, splice site and nonsense mutations, or variants with a high evidence as outlined in the guidelines. If, based on the evidence we have, no safe call can be made, the mutation will be declared as variant of unknown significance or variant of uncertain significance, VUS. And this will also be included in the report as such. So we'll ask the physician, for example, to provide germline material such as nails or hair or buccal swabs of the patient for further clarification for this respective patient. And the information will finally be added to our in-house database as well to be better informed at the next patient. If we now combine all the different tools that we have from databases worldwide and from our in-house database, this is our interface for the interpretation of variants right now. It is based on a variant. All relevant information will be pulled from a variety of sources to help make the call for benign or pathogenic. This is an example of a variant in TET2. An entry exists in cosmic pointing towards a somatic mutation. No entry is in dbSNP, which is a collection of known SNPs, also pointing towards somatic. Now, dbNSFP is a database developed for functional prediction, and it is a compilation of 20 different prediction algorithms. Each of these tools use a different approach, such as an evolutionary conservation, protein structure stability, and so on, with their own strengths and weaknesses, and a high error rate. Thus, a consensus rating of multiple tools is used. As this is a very difficult task, we trained, in addition, a machine learning algorithm, which was fed with the classification data from the MLL scientists created over the last 10 years. In theory, we should receive a consensus classification recommended with the same accuracy as all our trained MLL scientists and all available databases in combination. In addition to the publicly available sources, data from our LAMP system is shown as well. On the left bottom, you see the respective TET mutation that we found in this patient. We used the aggregated variant allele frequency as a metric to distinguish between mutation and SNP. The reason is that typical SNPs will be presented with either 50 or 100% variant allele frequency, whereas somatic mutations are below 50 or between, let's say, 52 to 99. Now, this is the workflow for today as we have implemented it in our routine. And you see here from all the different techniques that we use to combine the best results to diagnose, to classify for hematological diseases, starting from cytomorphology and ending today in a routine setting that is completely accredited according to ISO 15189 with next-generation sequencing. And you also see here the respective turnaround times for NGS in-house. Now, having said all that, it seems that this was a straightforward enterprise that we took, and this is not true because it sometimes looks even more like that. We finally hope to see the top at the end of our ideas and starting to work with new assays in a research situation and bringing them hopefully as quick as possible from research to routine. And the question is, how will this look in the next future? Now, the question is, would be, for example, exomes be successing the panel testing? The input is a few, only 100 nanogram of genomic DNA, and you will hopefully see all the coding regions. The coverage can be 100. This is less than we normally achieve by panel sequencing, where the minimum coverage we use is 400 and the medium is 1,800. But there are a lot of pros, especially if you compare this to the whole genome sequencing. It's cheaper and you have a better coverage and a higher data quality. But if you think about, will this be better than panels or picking up translocations by assays we use right now? Although the exome is only 1% of the complete information, you will not see structural variants such as copy number variations or loss of heterozygosity. There will be maybe also incomplete coverage of all exons. And the question is always, this is also true for the panels, what are we doing with the data that were not requested? How do we store them? How do we work it out? Also from the ethical point of view. Now, I would like to share with you what we think maybe one phase between the panel sequencing we use right now and the future of diagnosing leukemias and lymphomas and other diseases. And to learn about the options, we started a cooperation with Illumina as well as IBM Watson this year in March. And the intention to do this is to use a large-scale approach, investigating a lot of genomes and RNA-Seq, and to address this data set with the best available software tools to, we call it the moonshot for MLL, that is only possible by combining all our efforts with Illumina and IBM Watson. And the vision we have is to improve the diagnosis of leukemias in the future, to even refine classifications that in many diseases is still based on the phenotype, to guide targeted treatment that is more and more possible, and also to see genes or mutations that can improve our strategies for minimal residual disease testing. And using all this together, the fusions, the single nucleotide variants, the expression for entity-specific aggregation. Now, I would like to hand over to the poll question. Svetlana, you want to take it?
Yes, thank you very much. So we have one question for our audience. So when considering the adoption of NGS for whole genome sequencing, what is holding you back? Please select the most relevant response. Cost of sequencing, bioinformatics infrastructure, lack of clinical evidence, missing expertise for analysis. So again, the question is, when considering the adoption of NGS for whole genome sequencing, what is holding you back? Select the most relevant response from below. Cost of sequencing, bioinformatics infrastructure, lack of clinical evidence, missing expertise for analysis. So we have the answer. So all apologies. So it seems that the cost of sequencing actually was around 40% of our audience that found as one of the biggest challenges. So Professor Haferlach, is there a comment that you would like to make on these results of this whole question that we just did?
Yeah, thank you very much for being part of this presentation, because I wouldn't know what I have to answer here, because all four may be of importance, and so I see where you think are the most difficult problems to be solved. That means the costs and the bioinformatic structure as well. Missing expertise for analysis, for sure. I'm, let's say, surprised, maybe I say I can be happy, that only 12% of you say lack of clinical evidence. So if you would have the option to vote for more than one, you may think about it different. But I would say I can follow what you just addressed in these answers. And let's look here what will be about the costs. Now, this is the slide I borrowed from Illumina. It's the only slide I borrowed from them. And you see here the cost is going down over the last 15 years. Now, we started with $3 billion per genome between 1990 and 2003. And I have to admit, depending on the number of cases you analyze per year, the costs are going down to $1,000 per genome. This was the eye-catcher 3 years ago. And I have to admit also that these costs were calculated if you cover genome 30x. And I will tell you why this is of importance from our perspective for what we are doing right now with our moonshot. But anyhow, it seems to be possible that if this goes on a little bit further, there may be the option to use even whole genome sequencing in a diagnostic setting in the future. And we, at that time, will not have covered the problems with bioinformatics and how to analyze the results and bringing them to the report. And I also admit that the clinical relevance has to be proven. Now, we want to start to prove this, to prove the clinically relevance. And what we do in the project together with Illumina and IBM Watson is we use the NovaSeq. This is the new high-throughput sequencer that came to the market in the first quarter this year. And it opens a lot of options in one instrument. The runtime for a genome is 40 hours. You come to a huge number of terabases sequence. One important aspect from my side I see is that there will be 3 or even 4 flow cells, and the applications are different, like genomes, exomes, amplicons, or MRD as well, transcriptomes, targeted RNA, and even liquid biopsy will be possible. And as the flow cell in comparison to the HiSeq from the NovaSeq looks like that, the different lanes are there in the next future also to address different assays per run. That means you can run in the next future in one lane a DNA-based assay and the other one the RNA-based assay, and the third one, let's say, the MRD samples you want to measure. And that makes it very flexible for research, but also for routine use in the future. So what we are doing in this project looks like that. So to run the project in a time that we would like to have it ready, we use right now 5 HiSeq-X and 5 NovaSeq in parallel in our lab. We can run 5 samples on the HiSeq-X per run and 6 samples on the NovaSeq per run per machine. And we cover all these genomes 90x minimum. And in parallel, we do on the NovaSeq also the RNA-Seq, 64 samples, 2 runs a week, summing up for, right now, 130 cases per week that are sequenced. And if we are going to start to use the S4 in the next 4 weeks, that will be increased to 200 samples per week. Now, we have a lot of information now available by doing so. And the NovaSeq and the HiSeq are connected to the base cloud in Frankfurt. And we have a hub there that is at that time given and owned by the MLL and is constantly streaming the data as they are being produced by the sequences. Subsequently, to the completion of the run, the generation of the FastQ files is started automatically producing demultiplexed files for each of the samples in the run. And the demultiplexed files are used then for the following alignment steps, which each read in the FastQ is assigned to its position in the HD19 reference genome. The line data is subsequently used for variant calling. And due to the lack of normal material, we use a so-called tumor unmatched normal workflow, where our tumor samples is subtracted against the reference DNA which was sequenced along the sample to eliminate sequencing specific artifacts. This results finally in a VCF file, and this contains all variations in the reads against the human reference genome, and it serves as the input file for our collaboration with IBM Watson to apply artificial intelligence and to interpret whole genome sequencing data. And the VCF is also the starting point for our analysis. And the next slide shows you the base-based workflow we use here in-house to run all this data. The primary tools we use is the base and the variant interpreter and the code analyzer from Illumina. We import the VCF files to the variant interpreter first, and due to the unmatched normal workflow, the VCF file consists of around 4 million putative variant entries per sample. That's a lot. So the variant interpreter is a software to filter and visualize the sequencing data, particularly from whole genome sequencing and from RNA-Seq. And our application is the comparison of data obtained with whole genome sequencing to our orthogonal data, which we acquired during our routine operations I showed you at the beginning of the talk, such as cytogenetics of fish or standard PCR or even targeted NGS sequencing of specific panels. And the interpretation in the variant interpreter is case by case, whereas for an aggregated view to the data, we also import the filtered VCF files to the cohort analyzer. And by doing so, we, in the last 6 months, analyzed and investigated 2,000 genomes and transcriptomes that are completed, so we are ahead of our schedule because the end of our project we schedule for the end of March next year. Now here's one example. So this is a CITES picture where you can see that this patient has a very, let's say, complex karyotype. That means you see the 1517 translocation, which is a typical translocation in acute promyelocytic leukemia, but you also see that is also translocation 817, and you see derivative chromosomes 8 in this case. So this is from chromosome banding analysis and from 24-color fish. If we now have a look for the readout that comes from the variant interpreter, and the same case is shown here, you very nicely can see On the upper level, the translocation 1517 and 1715 because it's reciprocal. And you also can see the 817. And on the middle level, you can see the derivative chromosomes 8 with gains of chromosome 8 being shown there. This will not be enough to go case by case into the data. And therefore, the only way to analyze this vast amount of data generated by whole genome sequencing is to apply methods for which an increasing amount of data is not a curse, but it's a blessing. And the majority of these methods belong to the field of so-called artificial intelligence. Methods from this field, especially deep learning algorithms, such as IBM Watson is using, improve their performance by learning from experience. Hence, the more data is thrown at the algorithm, the more accurate is its prediction. So neural networks and other deep learning techniques, as depicted here on that slide, have the additional advantage to be able to recognize underlying patterns, rules, and even principles directly from the data, mostly without human intervention. Now let's have a look here. This is just a picture of what the readout that we finally hopefully can understand a little bit again may look like. So patterns can be basically everything. And the comprehensive data obtained from whole genome sequencing might allow us to draw chromosomal maps, starting with chromosome 1 left up, ending with chromosome 22 and X and Y in the down right corner, and showing all these occurrent somatic variations which might be able to differentiate not only between healthy and tumor samples, but also between various disease types such as shown here, MDS and AML. Moreover, these genomic profiles could be used to train neural networks, which might further aid the diagnostic procedures in the next years. Now, we already use neural networks to distinguish between AML and MDS, and the accuracy is up to 93.5% already based on the data. If you just use mutations, 88%. If you combine mutations with cytogenetics, 93%. But if you look into the transcriptome, we reached so far by our neural networks 93.5%. Now, combining all this with the knowledge we have already from the patient, from our standard approaches, we can set up a landscape of translational medicine by age of the patient, by blast, by response to treatment, even by the history of the disease, and adding the RNA-Seq data and the whole genome sequencing data into these bioinformatics tools, statistic tools, and artificial intelligence, leading to the best possible specific diagnosis. This is at least our goal and our vision for the next years, and serving the patients with his specific individual treatment and also recommending MRD marker. So we face a very broad spectrum of instruments, of software. We need to have in mind the turnaround time, and we had to write the reports. But this is not all. We need to have taken care for diagnostic accuracy. We want to have targeted treatment. We need markers for MRD. We need to be always within the classification rules that may change in the next years by approaches like that. And we want also to support drug development. Now, if you say, well, that's far away from my routine, my world looks different, I have to think about it, and maybe this is true in the next 10 or 20 years, I just want to make you aware of the world you are living in right now. And even if not whole genome sequencing is the today's world you use to diagnose leukemias and lymphomas, this is the world you live in right now. And this is a world based on tools that change our world, sometimes worse, sometimes better, but it's there, and it changes every week. And if we would just have in mind that this is the reality, especially for younger people in their daily life, in their routine they live. I think we have to also have in mind that the approaches we have to do are global approaches. This is not a single institution approach. This is a global approach to diagnose the best way we can do. This is centralized. I have to say that, I think, in the future, at the beginning at least, and the readouts will be cloud-based. We have to think different. We have to think globally, and we have to think using tools like artificial intelligence. So let's conclude for this talk. My conclusion for today would be NGS is the new gold standard of sequencing in hematology, because in most cases more than only one gene needs to be investigated. Now, doing all that in a huge number of patients per day in a mostly, hopefully, automated workflow in the future, you need to be adjusted for your panel testing right now, and you have to also be there with a very short turnaround time to serve the patient's need, because an acute leukemia needs a very short turnaround time. The use of whole exome sequencing or whole genome sequencing combined with RNA-Seq needs to be defined, and what we are doing right now is to do a first approach to compare it with a routine diagnostic approach as state-of-the-art right now, and find the gaps, but also the needs and also the options of tools being NGS-based to be implemented, augmented by artificial intelligence approaches in the next years for diagnosis in leukemias and lymphomas.
Brilliant. Thank you, Professor Haferlach. With this, I would like to end this webinar. Thank you, everyone. Thank you, Professor Haferlach.
Thank you very much.